Hello and welcome to EJC News Focus. If you were designing a new cancer drug, what criteria would you want it to meet? That it increases DNA damage perhaps, or reduces DNA repair? That the target enzyme is not essential to normal cells and is druggable, or indeed all of these? Thomas Halliday from the Karolinska Institute in Stockholm has been working on MTH1. He believes this work will lead to a compound which ticks these and many other boxes. I asked him to tell me more.
So I wanted to challenge the paradigm of personalized cancer therapy, that we should target the things based on the cancer genotype. So rather, we wanted to find a way to target cancer more in general. And so the criteria that we needed to meet then, because we still wanted to do a targeted therapy. So one criteria was that we should cause DNA damage to kill cancer cells. So causing DNA damage was important, and also we didn't want to kill normal cells. So therefore we want to exploit that cancer cells have a high level of already present of DNA damage. So the cancer cells already have the potential curative lesions in themselves. So we want to exploit the fact that they already have the lesions. So by doing that, we should also inhibit DNA repair. And then, of course, we want to target a non-essential enzyme. And this is important because we don't want to have any side effect associated with the targeted therapy. But it needs to be then essential in cancer cells. And finally, the final point is that you need to have it druggable. So you need to have a target that you can actually make small molecules and that you can inhibit, because there are some proteins that are just too difficult to make drugs to.
It sounds almost impossible that a drug could fit all of these criteria.
I've done a lot of RNAi screening and screened the big libraries, but I thought this time that there's also a lot of information published. So I went through did my homework, you could say, and went through the literature and found very interesting, compelling data suggesting that this is a very interesting enzyme to target.
What was the enzyme?
So the enzyme that we found is called MTH1. So this enzyme ensures that the building blocks that are normally damaged before they are incorporated into the DNA are never incorporated into the DNA. So they are sanitizing the damaged nucleotides pool, you could say. So the nucleotides that build up the DNA. So they're ensuring that only the correct ones can be incorporated into the DNA molecule. This is not only working for a specific cancer type. This is working across every cancer. So it's working, but not for normal cells. So it's specific for the cancer phenotype, for the cancer cells. So we call this cancer phenotypic lethality, that it's only lethal to cells when they become cancerous.
How has the research progressed?
We have managed to really prove that this enzyme is required and essential for most cancer cells, and not essential for normal cells. So that was one of the criteria, and we can show then how the cancer cells are killed when we remove this enzyme. And furthermore, then we had to develop molecules, small molecule inhibitors, to target this enzyme, and that is not trivial because this belongs to a new protein family that has never been drugged before. So that took a big effort for us to go into medicinal chemistry and to make the small molecule compounds, optimize them so they can be used in animals and to be used to really target the cancers. And now we're at the stage where we can scale up and start clinical trial.
You also found the compound is particularly effective when cancer cells have developed resistance.
Yeah, so normally today cancer patients die because of resistance to chemotherapy such as cisplatin or Taxols. So the reason why patients actually die is that you cannot give more chemotherapy to them because they are fatigued. They are just too tired to be able to take the side effects from the chemotherapy. So we can show that our compound is selectively killing the chemo-resistant cells. So we are selectively killing the cisplatin and Taxol-resistant cells. So this is very encouraging for the late-stage patients, that potentially we can kill off their multi-resistant cancers without causing the side effects that they normally have.
When will the clinical trials start?
So clinical trials will start towards the end of 2015 or potentially the beginning of 2016. The patients that will be involved in the clinical trials will be all comers, so it will be a lot of different patients because it has broad activity. And the primary purpose of the first clinical trial is to look at the safety profile of these small molecules. And then, of course, also we hope to see that we can see some anti-cancer activity.
You want this compound to be developed through open innovation. What exactly does that mean?
So yes, we like open innovation, and what we mean by that is that we are embarking on something novel. And novel also means that it is complex and there is a lot of things that we need to understand. And we are not a company, so we don't keep the target secret, we don't keep the structures that we have made of our molecules secret, we keep it all open. And also scientists across the world, rather than keeping it secret to them, we send it to them for free. So we give out these compounds for free for them to try with their own ideas. And they don't even need to tell us what they want to do. So we don't want to control the information.
Has this model been used by other groups?
So this model is quite common, open innovation, when it comes to just academic research. But it's not very common when it comes to drug development. And the reason is that a drug company wants to ensure that there is no unfavorable information that becomes public about their molecules. And I can see this point, but I'm taking it from a different standpoint. I'm taking if there are some unfavorable information about these molecules, it's good if we know them up front. And if it's public, well, we have to deal with it. And we cannot keep things secret. We have to do things in the open. And that is also giving us credibility. And this is also a reason why we have started a foundation. So all the intellectual property and all the everything is then belonging to a public foundation, which means that if there's any benefit to this, we can also give back this to medical research rather than to investors and to bankers, and the benefit from this therapy will go back into cancer research in the future.
So a new approach for a new compound, and one we'll expect to hear a lot more from in future. And with thanks to Thomas Halliday, that's it for this month's EJC News Focus.